Well, as more people are coming in, I'd like to welcome all of you to our talk today that will be entitled Genital Herpes Vaccine Development, the Friedman and Cohen Labs Approach at UPenn. And before I introduce Dr. Friedman, I just wanted to remind everyone, Herpes Cure Advocacy, we are a grassroots volunteer organization that is not very old, but over the past few years that the organization has existed, um, which really has one mission, which is to cure, treat, and prevent herpes infections, has been able to do a tremendous amount of work, both on policy and a number of other areas. So certainly donations and support of the organization is always appreciated. And for today, I get the honor of introducing Dr. Harvey M. Friedman. Dr. Friedman is an infectious disease physician at the University of Pennsylvania. He obtained his MD degree from McGill University in Quebec, Canada, moved to the US after his residency training in internal medicine at McGill to do a fellowship in infectious disease. He's been at UPenn ever since, working as a physician and scientist. His time at UPenn included serving as the head of infectious disease for over 2 decades. And most importantly for today, his research focus has been on herpes simplex virus. Early in his career, his lab developed two novel mechanisms, which he'll speak about today, by which the virus evades our immune system. He has and has gone on to develop that, has had a new focus, which was on assessing if these newly discovered mechanisms could block the virus from entering using these escape mechanisms for vaccines. Blocking immune evasion is a novel approach for vaccines design, leading BioNTech SC, a German biotech company, to license the HSV vaccine technology from University of Pennsylvania. He has continued to advance this concept into human trials for prevention and or treatment, the vaccine trials, which I think he'll talk more about today. So it's my honor and privilege to introduce Dr. Friedman.
Thank you, Ken, for that kind introduction. And I want to just reiterate what you said, how great the organization is. It's brought attention to herpes at the NIH level under a previous administration, albeit, but it's been a great voice for herpes vaccines, herpes diagnostics, herpes treatments, and congratulations. Okay, let's get started. So I work very closely in my lab with Gary Cohn's lab in the dental school, and I'm going to tell you what we're doing. I'm going to spend a little more time on this slide because of the current climate and in the U.S. and how many people have viewed vaccines as, anti-vaccines or and we only do it for profit. This disclosure. So I'm an inventor on a patent family that's been licensed by Penn to BioNTech. The patent family is being developed by BioNTech. I am an employee of Penn and don't hold any financial interest in BioNTech. The opinions you're hearing today are mine, not those of BioNTech. The candidate vaccine that we'll talk about more at the end it has not yet been approved for general public use by the FDA. Obviously, we're hoping it gets to that, but it's not there yet, and there's no guarantee it will. So the general outline of the talk is we're going to talk about concepts of vaccine development, past HSV vaccine trials, our candidate, and the status of phase 1 trials. Now, first, some of the general concepts for herpes vaccine, genital vaccine. Genital herpes is a sexually transmitted infection. Globally, 13%, that's a big number, are infected between ages of 15 and 49. That means it's more than 500 million people globally are infected with this virus. Now, some of them don't know it because it can be quiescent or asymptomatic. Women make up about 60% of that total, men 40, and that's anatomical. The anatomical differences explain that women are just more susceptible to the virus. And there's two viruses that can cause genital herpes, HSV type 2, which is the dominant cause. About 95% of the global population has type 2, or HSV1, which in US and other resource-rich country makes up maybe 30-40%, but globally it's only about 5%. And if people want to ask why at the end, we can go into some of that. But now HSV1 is the oral herpes. It can cause genital, but it mostly causes oral. When it causes genital, it tends to be less frequent in its recurrences than the type 2. As a cause of oral infection, lip, cold sores, 67% of the world population, 5 billion plus, has this infection. There's two goals for a herpes vaccine. One, to prevent infection, and a great example about a STI, sexually transmitted infection, that is prevented by a vaccine is the human papilloma vaccine that's used for cancer prevention. It's given to young people before sexual onset, before sexual activity. In the long run, if we have a herpes vaccine that works for genital herpes, we'd like to target that same age group. Or the vaccine can be used to treat people already infected. It doesn't have to be mutually exclusive. Ideally, a vaccine would do both, prevent and treat. But it may be one vaccine will prevent and a different vaccine will treat. Now, off to the right, on my right, the right at the end of the slide, there's a picture of the virus. The squiggly thing in the middle is the DNA. It's surrounded by dense protein. And you can think of this as your brain surrounded by your skull to protect the brain of the virus. On the surface of the virus are these push pins. And these are called glycoproteins. We're going to talk about glycoproteins. Some of these glycoproteins help the virus get into the cell. Viruses have to get into cells to reproduce themselves. Others are involved in protecting the virus against immune attack, or their immune evasion molecules. All right. Now, I want to review with you what the life cycle, what the virus does when it infects a human. So at first, this is the virus. These little red knobs are the glycoproteins, the squiggly, the DNA. So the glycoproteins allow the virus, they attach to receptors on cells, they get into the cells, reproduce themselves, hundreds of copies are made. The cells are the factory the virus uses to reproduce itself. It goes from one cell to another, and if it goes to enough of those cells, it will cause a big enough lesion, big enough area of damage that you can see it as a genital ulcer or a sore. It finds its way to nerve endings, and then it will travel along the nerve, shown in green, and travel up to the nerve cell body, which lies just outside the spinal cord for genital herpes. In those nerve cell bodies, the virus becomes dormant, or we use the term latent in nerve cells. And then it periodically reactivates, it's not dormant for very long, or sometimes it's dormant forever. But when it reactivates, it will travel back down nerve fibers to cause a ulcer, a lesion again, cause disease. So primary disease when we first see the virus, latency, reactivation, and then more lesions and disease later on. Now, where can a vaccine intervene? Well, I have an intervention here. Don't let the virus enter. And we'll talk more about that. Most vaccines that are out in use now use this as a strategy. Don't let the virus, not, now there isn't any vaccine for herpes, but most of them that we have for other viruses use this as their key strategy. Don't let the virus in. Viruses have to grow in cells. If they can't get in there, you win. The vaccine wins. Next is you can try to stop the spread from one cell to another. And that would be effective because even though you make it a local lesion, you will stop the development of latency. It never gets to the neuron cell body where it wants to become dormant. Or if it does get there, we can try to hold it there so it never comes back out again. Leave it there. It's not going to cause, well, we had the acute infection, but that's it. We're not going to get the recurrences, which are so troublesome. Or another site, we can have a vaccine intervention, or we can have all of these things. It's not mutually exclusive. If it comes back out, let's not let it make it, let's attack it with our immune system so we don't have lesions, ulcers, sores. It causes no damage or little damage. Now, how can a vaccine intervene? So that's what the virus does. And I showed places where we can stop it. So what does a vaccine fit into this story? Well, the vaccine stimulates our immune system to produce antibodies, these guys here, these Y-shaped molecules, or T cells, shown down here at the bottom, that can if the T cells are there to be prepared. So the host, the host is us. We can be prepared for the virus if it shows up. So a vaccine for prevention, we want to have that person, because we vaccinated them, to have these antibodies ready to go, have these T cells ready to go. So if the virus dares enter us, we can attack at all these different levels. If we don't get it to stop entry, maybe we can stop spread. We can hold it in its latent position, not let it reactivate. It's to prepare the host as best we can. So if the virus dares to infect us, we're ready. A good vaccine will be ready. Now, what's happened in the past? The past vaccine efforts at preventing genital herpes, there's been three large trials published in 1999, 2002, 2012. So it's a long time ago, 2012, 14 years ago. And we're still not there. Well, we're hoping we can move that needle and get there. So what happened in those? These F trials their goal was to produce antibodies that prevent the virus from entering cells. They targeted entry molecules on the virus. One of the entry molecules we'll talk about is glycoprotein D. This molecule is essential for the virus. Virus needs this to find its way into the cell. It binds to receptors on the cells, and it triggers a whole cascade of events that allow the virus in. It starts that cascade. Now, there's also two of these trials were aimed at just using glycoprotein D, the 2002, 2012. The other trial, the first one in 99, used two entry molecules. Glycoprotein D is the one that starts the whole process. Glycoprotein B, as in baby, is the one that actually allows the virus to fuse with the cell, so the DNA of the virus gets in and takes over the cell, and virus replication occurs. Now, I note here, each trial had some benefit. But none of them met the primary endpoint of the study. Some benefit In the first study in 99, it delayed onset of genital herpes. In 2002, it worked in a subset of people that were enrolled, women that had neither HSV1 or HSV2 prior infection. It worked in those, but that wasn't what the primary endpoint is, and the FDA said you have to do it again because it may have been just a fluke. And when they did it again, and this time in 2012, they only enrolled women that were double negative, never had HSV1 or HSV2, and it didn't work for preventing genital herpes. The FDA was right, but it did work for preventing genital herpes caused by HSV1, but not HSV2. So it worked partially. Each of these had some benefit, but not the intended benefit of the study. They were hoping for more. So what do I conclude? Targeting virus entry is helpful, but it's not enough. Okay, can we build on it? Now, past efforts, vaccine efforts to treat, that last slide was about preventing. In people with frequent recurrences, this is a therapeutic, a treatment vaccine. There's many failed efforts, unfortunately. Most of the effort have aimed at improving T cell responses, trying to, people already infected, the neuron cell bodies that are in those ganglia outside the spinal cord are already containing virus. They're trying to hold the virus in place. Don't let it out. Or if it does get out, don't let it cause any lesions at the recurrence site. And again, a few of these have showed limited benefit, but not strong enough that any company has wanted to take this to the market. It just hasn't been good enough. They didn't miss by a lot. So for those of you who are hoping for a breakthrough, we've come close on a number of occasions, scientific community, but no one has hit a home run yet. So why in the therapeutics have we not hit a home run? Well, because no one has figured out how to improve upon immunity that develops after infection, i.e., what is missing that a therapeutic vaccine can make better? These people are infected with the virus. They're getting infected over and over again. They're making an immune response, and yet they can't control the virus. And if we can figure out what's missing, we might be able to control the infection by using a vaccine to immunize with the missing piece rather than what's already being responded to. Now, what's our approach? Well, we're targeting three glycoproteins. Those are these push pins on the viral surface. Uh, one of these are all, okay, I want to make this point. These are glycoproteins are all expressed on the virus envelope, which is on the surface of the virus. And when the virus replicates or grows within a cell, the virus puts these little push pins, the glycoproteins, on the surface of the cell as it's replicating to protect the virus or the virus infected cell against our immune response. So they're surface expressed proteins and those are the ones, not all of the escape mechanisms the virus uses are surface expressed, but the ones that are amenable to the strategy that we've adapted, which is to try to make an antibody response to these proteins so they cannot interfere with the immune response. I'm going to show you some of that in a few minutes. So the three we have, one's called glycoprotein D. It's an entry molecule. The others are glycoprotein C and E. There's four molecules needed for entry. They're shown here. GD, as I mentioned earlier, gets it started. We picked GD because my colleague Gary Cohn in the dental school is an expert and had all the right tools to study this. And C and E we picked because of observations made in my lab over the years that about immune evasion. And we had the idea maybe we can stop the virus from using those tools. So in a little more detail and in a cartoon fashion, glycoprotein D on the surface of the virus, our goal is to block virus entry, as many other vaccines have done, but as I said earlier, it hasn't been enough. So that isn't our sole goal. It's part of our strategy. Can we block virus entry? It's a good strategy. We want to try to add to it. So here, these triangles are glycoprotein D. There's quite a few of them on the virus particle. They allow the virus to bind to molecules on our cell surface that are receptors for the virus. The virus binds, the cell lets the virus in. Big mistake, but it's done it. The virus takes over the cell and starts reproducing itself, making hundreds of copies that will spread from one cell to another. And in that model I showed earlier, eventually the virus finds a nerve ending, travels along that, develops latency. So what do we want to do? We want to have antibodies made in advance of infection. We're immunizing a person before they ever see the virus. This is a prevention vaccine strategy. And so they have antibodies waiting, daring the virus to enter. And if it does, bango, they're going to take care of that virus and not let it get into the cell. And it's a good strategy, but enough virions escape that it's not good enough. So what else are we doing? Well, we've added this glycoprotein C. It blocks immune evasion. Now this viral protein, glycoprotein C, we discovered in 1980s, so that's 40 plus years ago, that it binds to a complement protein. It's called complement C3b. So what's that? The complement system is part of our body's earliest response to a foreign organism, a pathogen, a bacteria, a virus that attacks us. The complement system recognizes that invader as foreign. And there's a whole series of proteins of which C3b is a central, absolutely essential part of that cascade of proteins. What it does, this is a picture of GC on you can picture this either on the surface of a cell, because I mentioned the glycoproteins get expressed at the cell surface, or on the surface of the virus. Both of those are correct. And what GC, the glycoprotein, does is it binds this central protein in the complement cascade. The complement is attacking the virus. It's trying to punch holes in the virus membrane that will kill the virus. It's trying to punch holes in the cell that will kill the virus. And the virus says, not so fast, buddy. And it binds C3b to turn that off, to block it. Because if you imagine, I have a little corner here that's bound to C3b, and you can picture it that this little corner is necessary for the next step in that cascade to activate the next member of the cascade that eventually is going to lead to the punching holes in the viral envelope. And the virus says, forget it, buddy, that's not happening. So what are we doing? Well, we're hoping to make an antibody to GC2, much like I said for GD, that binds to it and is ready to act if the virus dares to come in. This antibody binds to GC2 and it blocks the ability of the glycoprotein to interfere with complement. And we're hoping that will allow complement to work. Now, we discovered that mechanism back in the 1980s. If you look at the list of pathogens, microorganisms, viruses, bacteria, fungi that use a similar approach. It's not the same protein, but the same idea. Make a protein that inhibits the complement system. Look at the list here. There's a bunch of viruses here. Many of them are herpes family viruses. This is mpox is from smallpox family. There's important bacteria. There's yeast and fungi and parasites. All of them are making proteins that do much the same as what we have found with herpes. And so the conclusion I reach is that it's probably really important. If all these organisms are giving up part of their DNA to do interfere with complement, it's very likely that uh, protein is for the advantage of the microorganism and the disadvantage of the host. The bacteria are wanting to protect themselves. So it's an impressive list. Now, the third vaccine antigen in our vaccine is called glycoprotein E. It also is expressed on the surface of the virion, here, shown here in cartoon. Think of this as the virus surface or the surface of infected cell. It works in collaboration with another glycoprotein, glycoprotein I. The 2 means it's from type 2, or HSV2. HSV1 does very much the same things that I've been talking about. And now let's look at that GD antibody that we vaccinated with GD and the GD protein, the entry protein, and the virus has dared to come in. And now we have our antibody ready to go, and it's attacking the viral protein, not letting it enter. But it's what the virus is doing. Okay, you got me at the GD end. This is the antigen binding end. You got me. You got me. I admit you got me. The virus is admitting to being had. But this end of the antibody molecule is doing a lot of important things that help us clear a virus. And they say, okay, you may have gotten me at the front end, but the back end of the molecule, which is doing a lot of important helping our immune system respond to you, you're not getting that out of us. The virus binds to the FCN, that's just called the FCN, binds to it and blocks the ability of this part of the molecule to interact with the immune system. So you get something out of the antibody, but not everything. The virus says, okay, I'm going to mess up at least part of the antibody. Now, what we want to do, that's the same theme, have antibody available before the virus ever comes in. So when it does come in, it can bind to this glycoprotein and not let the glycoprotein interact with the GD antibody or any antibodies that the host that we make as a result of infection. If it gets started, we'll make different antibodies to other proteins on the virus, and we don't want this. Well, this antibody to GE will stop that from happening. That's what we're hoping. All right. Now here too is a list. Now it's not as big, but we found that, this is that mechanism of how the virus interferes with antibodies. We showed it for type 1 virus, for type 2. Others have shown CMV has a bunch of these proteins that do similar things. The chickenpox virus has a glycoprotein that does similar things. Some of the bacteria that are well-known bacteria, staph, strep. So again, these organisms would not waste their time or their DNA on uh, encoding these FC binding proteins. That's what we call them, IgG FC binding proteins. They wouldn't waste their time if it wasn't important for their defense. So that's encouraging to us that there's many organisms that do that. Now, we're going to we address three questions in mice and guinea pigs. Is the immune evasion from antibody and complement useful for the virus? So we said we want to stop it, but now I'm going to ask the question, is it important to the virus? I said there's a lot of other organisms that have it, but that doesn't mean it's important to herpes. And I'm just going to summarize about 10 years of work here and say that we made mutations in the virus, herpes virus, small little mutations in the evasion molecules, glycoprotein C and E, changed it a tiny bit, but showed that we changed it so it could not um, evade antibody and complement. It lost the ability to interfere with antibody and complement. The mutant virus was now 5,000-fold less potent than the parental starting strain. 5,000-fold. That caught our attention. So is it important for the virus to make these? I'd say, yeah, it's pretty important. If it can't do this, antibody and complement are really active against the virus. Now, that's one question. Now, okay, so is it important? We wanted to know what's important. Can we do anything about it? Can we prevent the virus from using, from entering cells and invading? How do we stop it? We're not going to, it's not the mutant virus that's going to infect people, it's the wild type, the strain that's out in circulation. We use the mutant to prove a point, but what's out there in the community is not a mutant because the virus wouldn't survive if it was the mutant. So can we prevent it? So this is where we get into the vaccine side. And the vaccine we're using is an mRNA-based vaccine. And my colleague, Drew Weissman and Kari Carico, who are at Penn, were talking about 7, 8, 9 years ago about their research with uh, a modified messenger RNA that looked super duper great for a vaccine preparation. They were using it for other vaccines. It ended up getting used for COVID and had a huge benefit, and it won them the Nobel Prize for their discoveries. Drew is in the infectious diseases. Drew is one of my recruits to Penn, so that's one of my claims to fame. It's something I'm really proud about. Um, in any event, I knew what he was doing. His lab was on my floor. We were talking all the time. And I said with my colleagues here in my lab, why don't we try making our vaccine using modified mRNA rather than what we were doing was just going with a protein to start with. So the mRNA makes the protein in, you inject the mRNA into the people, and the people the mRNA, once it's in us or in mice or guinea pigs, will make up the proteins, the GCGD proteins that will stimulate our immune system to make antibodies and T cells. So instead of putting protein directly in, we were putting mRNA directly in. And it worked really well. But we were putting protein, which was good. mRNA was hands down better. It's a really great technology, one of the best advances in the last decade. And I won't talk about the politics behind what's happening, but it's a shame. So what we did, we determined that a vaccine that was using mRNA to express these proteins, C, D, and E, uh, in mice and guinea pigs, it could do what I showed in that cartoon. It could make antibodies that would block entry, block GD, and block the ability of the virus to evade antibody and complement. So can we use a vaccine to prevent the virus from evading and prevent it from entry in mice and guinea pigs? When we immunize them, we can get antibodies made that will do what we wanted it to do. Block entry, block the interference with antibody and complement. Okay, now we've answered two questions. Is it important? Can we do anything about it? The third question, now that we know we can block it, is that going to be helpful? And the answer here again is yes. And the way we did that, we took the antibodies that are made by immunizing mice, guinea pigs, and put them back into animals before we infected them. So we're putting the antibodies in. The concept, you want the animal ready. But we're asking a very specific question. Can we block entry? Can we block immune evasion? So we took antibodies to block entry, antibodies to block immune evasion with GC or GE, and put select antibodies in and asked, do those antibodies protect? And the answer was the animals that got the antibodies handled the virus far better than the animals that didn't get these antibodies. So we've answered three questions. Is it important? Yes. I'll go down here. Can we prevent it from entering, at least the mice and guinea pigs? Yes. And is it helpful to the host? Is immune evasion important? Was that first question. The third one is it helpful for the host? All are yes. So the concepts we were following at least look encouraging to us in animal models. Now, I want to go next to what happens when we put the whole vaccine into an animal? So these are going to be preclinical studies in mice and guinea pigs. So first in mice. Uh, we immunize with our three different mRNAs. The mRNAs are again glycoprotein C, D, and E. When they get into us, our body, our cells in our body that take up this mRNA will make proteins to C, D, and E, and those will stimulate our immune system to make antibodies and T cells against those proteins. Um, so when we put the vaccine in mice and we infect the mice either with type 1 virus genitally, or mainly we're interested in type 2 because that's the most common, but we also looked at type 1. Does it work against type 1? Because it's an important cause of genital herpes, I said earlier. And I'm scoring these on a scale of 1 plus is some protection, 4 plus a lot. So how did it do it in these models when we vaccinate? So the mouse is ready for infection, and then we infect. We in humans you can't infect. You can't, it's not ethical. But in mice we can, and we have the mice ready to see the virus. We put it in, and how effective is the virus at protecting? We put the virus in intravaginally. These are female mice that we use for these studies. It's the best model. And it protects on a scale of 1 to 4, I gave it a 4 plus. It's really good. Can it prevent the virus from establishing latency? And if you can, there'll be no recurrences. The mouse model isn't a good one for recurrences, but it is one to tell us if we can prevent latency. We did great. All right, that's encouraging. What about guinea pigs? Here we're only studying HSV2. HSV1 is not very infectious, not very potent as a cause of genital herpes in guinea pigs. So we stick with HSV2. So here again, prevention. Can we prevent genital lesions? I'm giving it a 4 plus. It may be pushing it a little 4 plus, but it's at least a strong 3 plus, maybe a 4 plus. Now I'm looking, everyone who's knowledgeable about genital herpes will say, well, how about shedding? Because this is something you get infected and you start having shedding and you worry about transmitting to your intimate partner. And I want to look at shedding in two ways. Is it shedding infectious virus or is it shedding DNA? Now, the tests that are done in clinical trials for uh, most antivirals and some of the vaccine trials look at DNA. But in our guinea pigs, we're looking at both. And one human study has looked at both. But I'm going to tell you just keep your eye on these two lines. Does it prevent shedding of infectious virus? That's what's going to infect your intimate partner. It's great. There's almost no time that we see infection, not never, but almost never. If you want to know how we do against DNA, it's not as good, but the DNA is not infectious. It's the virus that's infectious. It's a piece of DNA that is, just not going to transmit. Um, and so the message here is it's more, I think we should be focusing more on virus shedding than DNA shedding as a marker of infectivity, a point that's something I want to push going forward. The DNA scares the wits out of people, but when we look for virus, now I'm looking in guinea pigs, others have looked in humans, but the number of studies are so small for such an important point. Now, what about treatment? We're very interested in whether the vaccine candidate we're studying will work for treatment. So if we infect the animal first, that's like you have an infected person, so lots of recurrences, and then we vaccinate, what happens? And we can reduce the lesions, the number of recurrent lesions, genital ulcers. I'm giving it a 2 plus to 3 plus rating out of 4. It's not reducing it to 0. When I say not, some animals are 0, but some animals have breakthrough still, but there's still many fewer than they may have had without the vaccine. What about shedding a virus? Yeah, it works very well to reduce the infectiousness of that shedding, that worry that you're going to transmit. And I hope that happens in humans. It happens in guinea pigs. The infectious virus is just not there. And what about shedding of HSV2 DNA? Not as good, but that's not what we'll try. That's a marker of potential risk of infection. Shedding of virus is the real thing. And even when we find it, there's so little there that it's not something that'd be potent enough to infect most individuals. All right, so strong preclinical results, but keep in mind animals are not humans. So uh, I want to just review quickly. I want to leave some time for questions and get to the rest. So to the phase 1 study. So we're also interested in whether the vaccine works against non-genital HSV1. Does it prevent oral infection? Great in mice. Does it prevent eye infection? Keratitis model, eye infection. Great in mice. Mice aren't humans. Can it prevent brain infection, encephalitis, a big problem with herpes type 1? In progress. I can tell you it does really well. Does it prevent neonatal herpes infection? We know it does for the type 2 virus. We're working on it for type 1. So we hope that a vaccine that is effective against HSV2 may also be effective against HSV1, or we may have to modify the vaccine so it's effective against both. But if it's not so in humans, but in animal models, it's looking really good. All right, how about the human phase 1 trial? Phase 1 trial 1A. These are for safety and immunogenicity. 1A, I'm going to tell you about a 1B and a 1C. So these are, again, phase 1 safety and immunogenicity. Now, BioNTech reported the results of the 163 vaccine. And I mentioned earlier that vaccine has the same immunogens that we're using, but slightly different manufacturing and so on. It's not exactly identical. They presented these results in October. What the entry criteria were for people that had no known history of genital herpes. They may have had it, but they didn't know it. It was too quiescent, too minimal, too yeah, it was a minimal infection. Ages 18 to 55. There were 48 subjects. There were 4 doses. And it was first BioNTech looked at a low dose a slightly higher dose, a considerably higher dose, and then the highest dose they could. Don't forget, they're trying to figure out safety, what is safe, and what gives you a good immune response. And they're going to pick the highest dose possible that has the fewest side effects. And I'll go over that in a minute. But the immunizations, there was three of them at time 0, 2, and 6 months. And then they followed each subject for 12 months after the third immunization. That's one of the reasons it takes so long to do these trials, because now you're a year and a half from the day you enroll a subject before you're finished that subject, and there's 48 of them that have to be enrolled. They don't have to be enrolled, they can be enrolled simultaneously, but it still takes a long time. So what about the outcome? Then this is in BioNTech's words. They asked me to keep this to their wording. So the vaccine was well tolerated at all doses and had an acceptable safety profile. It produces good antibody responses. ELISA is a binding antibody. A neutralizing antibody is one that prevents virus entry. It produced good antibody responses. We don't know yet whether it produced antibodies that block immune evasion. I'm hoping we will be able to test that in the very near future. And it produced these in people that had never had genital herpes. And even if you had genital herpes but didn't know it, it boosted the titers moderately. It produced antibodies that not only reacted with the type 2 virus, the type 1. So that bolsters our hope that it will work against both. And the antibody titers against the glycoproteins persist for at least a year, which is good. We don't know how often the vaccine will need to be made, but the longer the antibodies persist, the better. And then they said in 2025 October that safety and immunogenicity will be further assessed in an ongoing dose escalation study. So now that's happened. So that's what is in the public domain because it was presented at the infectious disease meeting. It hasn't been published yet, but it is what they presented. Now the next two are still in progress, but they're well along. So 1B, what is it? No history of genital herpes. Again, very much like the 1A. This is to expand on that part of the study, thinking about safety and immunogenicity of a prevention vaccine. And they had two groups of subjects. This one group was antibody negative, had no evidence of infection to HSV1 or HSV2, and the other group was either seropositive, meaning they had evidence of infection to either oral herpes or it could have been general. You can't tell by the blood test, but HSV1 positive and or HSV2 seropositive. So antibodies. So think of it. This is a naive type subject. They'd never had a herpes infection of type 1 or type 2, and the other group had either 1, 2, or both. And this is fully enrolled now, and the data from this study will be rolling in as we talk. And they'll have an expanded number of subjects to tell whether the antibody responses, and they're also looking at T cell responses, are good. How good are they? And I'm hoping we'll get to look at do some of those antibody responses. They're going to look at it also. Some of those antibody responses do the things that I talked about. Block immune evasion from complement, block immune evasion by binding the Fc end of antibody. And if it does those things, we'll be very encouraged that we're on the right track. Now, there's another one, phase 1C. It's also a safety and immunogenicity, but now we're looking at subjects that have frequent recurrences of genital herpes. It's not a therapeutic vaccine trial. It's a trial of safety and immunogenicity in people that have frequent recurrences with the thought, well, we have to first know it's safe and immunogenic before we decide what's next. And so these are people that had 3 to 9 outbreaks annually. They're dosed a little bit different than the other one at 0, 1, and 2 months. It's also fully enrolled, and that data will be rolling in later in the year, towards the end of the year. When all of this is available to BioNTech, they're going to decide next steps. What's next? If these look good, then that will be very encouraging. So, summary, the RNA vaccine is doing what we hoped in the animal models. It's producing antibodies that block entry and immune evasion. In mice and guinea pigs, it shows strong protection against virus challenge with the herpes viruses. In phase 1 human studies, the 1A, it was encouraging, I'll say, and what we see in 1B and 1C will help determine the next steps. Do they go on to larger studies, or do they stop there? Let's hope not, but we won't know till maybe early next year. And then ongoing, what are we doing in the meantime? We're using our mice and guinea pigs to help inform decisions about what can we add? Does it work against HSV1? Can we add things? How do we get better T cell responses? Are they good enough? We're using our models to answer as many questions as we can. This is my acknowledgement slide. There's some of my lab members, current and recent. A lot of really talented people. They're showing up there. And the Gary Cohn's lab that has more than the 3 listed here because of some of them are students that are working in the lab. In this group, there are probably 8 or 9 PhD scientists. In this group, the total years of experience working with herpes is probably 300 cumulative years. It's just an amazing amount of experience that we've accumulated. It's never bad to have a Nobel laureate working with you. Our funding is from NIH and BioNTech, and I want to add from private donors. And if you're interested in donating, it makes a difference. We buy equipment, we hire more people, we buy supplies, we bring on more people, more equipment, move things faster. And so if you email me, hfriedma, no N, hfriedma, Penn Medicine, UPenn EDU, I'll put you in touch with the development office. We've had good luck with raising money, and it's made a big difference, especially in this climate where you don't know what's next in terms of when the axe might fall. All right, so I think with that,
On behalf of Herpes Cure Advocacy, we'd really like to thank those that attended, and especially Dr. Friedman for his work in the past, present, and hopefully in the future. Thank you so much for joining us today, and thanks, Dr. Friedman. On behalf